Welcome to NewYorkBIO's Virtual Breakfast Series, a digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode features Dr. David Fajgenbaum, a groundbreaking physician scientist, disease hunter, speaker, and best-selling author of Chasing My Cure. Best known as the doctor who cured himself of the rare disease Castleman's disease, and then founded the Castleman Disease Collaborative Network.
Good morning, everyone. My name is Jennifer Hawksland, and I'm the CEO at NewYorkBIO. Welcome. We're thrilled that you're joining us today for another installment of our Tuesday morning virtual breakfast series. Some of us have our coffee. If you do, that's great. We'll drink it with you over the next hour as we have very interesting conversations. And Derek, I think we may be starting a trend that I think that this is our third week in a row that our guest has been a published author.
Yeah.
We're giving our community a lot of extra bedtime reading over the next few weeks.
Well, as the kids go back to school, people are going to need book recommendations. So we've
there's not
filled
enough
in
to
on
do with everyone.
Yeah, exactly.
Homeschooling as well as doing our day job. So thanks again to our friends at Goodwin Procter, the law firm, for sponsoring our breakfast series in July and August. We really appreciate their commitment to our ecosystem in New York. And I will turn it over to Derek to introduce our guest, and then we'll get going with some Q&A.
All right. Thanks, Jennifer. So usually the way that we kick these things off is, I ask the guest for a little bit of an origin story and tell me how you got where you are. But, David, a lot of this story is really about how we got to where we are today. So we're actually going to go back a little bit further than that. We don't really want the 5-minute version of how we got to where you are, but let's take things back a little bit, and I'll set the stage for the audience. So you were in medical school. You were a Division I quarterback. You were an all world-class athlete, and you got a diagnosis that nobody quite understood. And you want to take us through a little bit of kind of the beginnings of the story, and we'll get into things.
Sure. So first off, thanks so much for having me on this morning. It's really a pleasure to be with you guys. So I was a healthy third-year medical student. I was at Penn, where I was training to become an oncologist. I wanted to treat cancer patients. I had lost my mom to cancer a few years before, and out of nowhere, I just started noticing fatigue that I'd never really felt before. I noticed like lumps and bumps in my neck that I didn't really know what they were, fluid accumulating around my ankles, and just this terrible abdominal pain. And as a medical student, we're all tired. So I was like, it must just be because I'm working really hard. I don't know what's going on. But over the course of the next couple of weeks, all the symptoms just got worse and worse. And I eventually went to the emergency department at the same hospital that I'd been working at. And they ran some blood tests, and they told me that my liver, my kidneys, my bone marrow, my heart, and my lungs were all shutting down. And so I was hospitalized right away and admitted to the intensive care unit.
Okay. And you basically, as things went, you had a number of cytokine storms. And how long did it take people to figure out actually what you had?
So
right, because you went through chemo a couple of times, didn't you?
Yeah, from the moment that I was admitted to the hospital and then the ICU, it was about 11 weeks of being in critical condition before I was eventually diagnosed. During that time, I gained 70 pounds of fluid because my liver and kidneys had shut down. I had a retinal hemorrhage that made me blind in my left eye. I spent literally weeks in and out of consciousness, and said goodbye to my family. And thankfully, right around the time that a priest came into my room to read my last rites to me, so when we thought it was all over, was when the diagnosis was finally made of idiopathic multicentric Castleman disease. IMCD is this rare immune system disorder where, as you said, there's a cytokine storm. Your immune system attacks your body's vital organs. Your immune system, which is supposed to be doing good and protecting you, actually attacks your organs. And at the time, so little was known about the disease, and it's called idiopathic for a reason, because we don't know very much about it. And as a result, chemotherapy was really the only option. So I got chemo, and it saved my life. It kind of was a last-ditch effort, saved my life. But unfortunately, as you said, I went on to have relapse after relapse after relapse.
Right. So, but in the process of those relapses, really where, some of the genesis of the story comes in, right? Because, in a sense, it gave you the focus to say, well, I'm going to figure out what's wrong with me, and I'm going to figure out, really how I can treat it. Right. So, I think there's an image that everyone gets, unfortunately off of TV. It's an episode of House, right? And it takes about an hour, and you start with a whiteboard and you start crossing things off the whiteboard. And after about 53 minutes, you have what you think it is. And then at 57 minutes, you get what it really is. And that's how it's solved, right? Is that, is that not the way it works?
That is not the way it works. But I do love House. But no, for in my case, it was relapse after relapse after relapse. But actually, with each one of those deadly episodes, and I've now experienced 5 of them, with each one of those 5 deadly relapses, I learned a lot about life. I learned lessons that I wish that I didn't have to go through what I went through to learn them, but I learned lessons that they fundamentally changed who I am. I mean, my life as I live it today is completely different than it was before each one of those episodes. And so, like I said, I would happily, give it away and say, I wish I didn't have to go through that. I wish my family didn't have to go through it, but some really, really, really key lessons emerged from those experiences.
Yeah.
Lessons
what are some of the biggest things?
Sure.
Yeah.
So the first one that really comes to mind is this concept of overtime. So you mentioned I played college football, and I played at Georgetown, where we lost a lot more games than we won, and certainly a lot more than went to overtime. But occasionally we won, and occasionally we did go into overtime. But what I learned about the experience of overtime is really that it's one where it's time you didn't think you would have. It's time where you really have to make the most of every second. If you make a mistake in the first quarter, you can make up for it. Make a mistake in overtime and the game's over. And that can be terrifying, right? I mean, that's actually a really scary situation, but it can also be really, really clarifying. So you can basically say, okay, I know the clock is ticking, but because the clock is ticking and because I'm aware of it, I'm going to make the most of the time that I've got. And, it's a cliche to say, live like you're dying, or live like, every day's your last, but I literally can hear the clock ticking. I mean, I know that I have this deadly condition that can relapse at any moment. And like I said, I wish I didn't have it. But this sort of setting of overtime, and it's a lesson that I try to encourage other people to think about their lives in overtime. And it's not to be afraid, but it's to realize that actually we're all in overtime. We all don't know, how much time we have. And as a result, it's not that we should be afraid every day, but it's that we should live with clarity. Like, hey, I'm in overtime. I'm going to make the most of every second. Because I actually made choices after the first time I nearly died that were different because I just gone through that. I was like, you know what? I just went through this. Like, I'm in overtime. I'm going to do the thing that I want to do. I'm not going to put it off to the future. It actually changed me fundamentally. So that's one. Another one that I like to think about a lot is the importance of humor during really tough times. I mean, you'd think that when you're in the ICU, that the last thing that you would want to do is laugh or smile. But I actually found that with my family, that during really, really tough times, even while I was in the ICU, if there was something to laugh about together, then that really helped us to get through the tough times. There was one experience on New Year's Eve of 2010 where my liver and my kidneys weren't working. So I had this huge belly because I just fluid everywhere, and I had a mask on. My head was bald from chemo. And I was walking around the hematology oncology floor with my dad at about 8 p.m. And we passed a drunk guy. He'd been clearly drinking on New Year's Eve. And he fell onto the ground, and my dad helped him back into his chair. And he looked at my dad and and he said, "Thanks so much. Good luck to you and your wife." We're like, "Wife? What is he talking about?" I realized he was talking about me. He thought I was my dad's pregnant wife, which we actually laughed about. And I mean, you could imagine, if you heard that, if anyone gets confused as their father's pregnant wife, that's probably something you could cry about. And it's probably something I would have cried about in maybe a different scenario, like, oh my gosh, this is awful. But I actually laughed about it because I was just so happy to be alive, and just being able to laugh with my dad was something that was really, really important for us. So those are just a couple of the lessons that come to mind. I mean, there's many more I'd happy to go through, but they they just have changed who I am. And that's why I wrote Chasing My Cures, because I don't want other people to have their last rites read to them and, you know, almost die 5 times to pick up on these lessons.
Yep. So one of the other things that I looked at here, there's a really great quote from one of the doctors that you ended up collaborating with, right? So to fast forward a little bit, you basically have yourself as, an N=1 patient,
Yep.
right? And, in addition to the things that you just mentioned, one of the things that happened was you had this dedication to taking as much data on yourself as you possibly could,
Yep.
right? And I believe the quote from one of the researchers that you worked with was, I've seen patients take data on themselves before, but I had never seen anything like this, right? And he describes, multi-tabbed spreadsheets and just a steadfast dedication to everything that you could measure, right? And I imagine that was another thing where you said, well, we're in overtime now, so
Yep.
I'm going to double down. I'm going to do every single thing that I know that I can do. And I suppose some of that started with thinking about data.
Yeah, there was a massive turning point for me, and that was that after my 3rd deadly flare, I'd now spent almost 6 months hospitalized in critical condition. With that 3rd one, I got a combination of 7 different chemotherapies that turned everything around. I mean, I literally went from like the brink of death to like back to life. And then I was on an experimental drug that was supposed to keep me in remission. It was the only drug that's ever undergone a randomized controlled trial for my form of Castleman disease. And so it was kind of the Hail Mary we'd all been hoping and praying for. I was like, oh my gosh, like this is amazing. Like the first randomized controlled trial is enrolling people right now while I'm like battling this disease. This is the prayer that, that we'd been hoping for and that seemed to be answered. And then I relapsed a year later on that drug. And so the drug works really well for about 1/3 of people, but I turned out to fall into the 2/3 where it doesn't work.
Right.
And so there was this really important point before I really dove into my data where I realized I was kind of on my own. I mean, I had a conversation with that same doctor where he explained to me that there were no more drugs in development, there were no more promising leads, and there were no researchers anywhere in the world doing work that was likely to result in any sort of progress in time to save my life. I mean, he just put it out there bluntly for me. And in hindsight, I'm so thankful he did, because he was so clear to me about how much work needed to be done. That made me realize that, like, okay, I'm on my own. The cavalry is not on its way. I can't just hope and pray that the cavalry is going to show up. If I'm going to survive, it's because I'm going to figure out a way to survive. I'm going to make it happen. And so that's when I just dove into data. I became a scientist. I mean, I was a medical student, but I had not ever performed laboratory research before. And I found a collaborator at Penn who let me start using some space in her lab and started doing laboratory work. I started a foundation called the Castleman Disease Collaborative Network. And really the work we did was based on 2 really important premises that I think are relevant for your membership and for people listening. And one of them is the critical importance of utilizing all the data I could get my hands on. I mean, I didn't have the luxury of being able to rely on randomized controlled trials of 10 different drugs and which one's going to work, which one's not.
Right.
I mean, the only drug wasn't working for me. So there was no other data that I could just find online.
Right.
I mean, it was about harvesting and getting all of the data I could on myself. And then it also was under the premise that there was no way I was going to develop a new drug or identify and develop a new drug in time to save my life. I mean, that's just that sort of a time frame is completely unrealistic. But what I did realize was that if there was a drug that was already FDA approved, that was already out there, and if I could use my data to find a pattern in my data that could suggest that an existing drug could save my life, that was the only shot I had. And of course, there was no way to know whether that actually was the case. I mean, it could have been that I searched and that no existing drug actually did exist. Or even worse would have been that an existing drug existed and I just missed it. And that, like, I picked the wrong ones. And in hindsight, actually, I did pick the wrong ones a couple times, but the 3rd one that I began testing on myself turned out to be what saved my life.
So how do you get? How did
you go about? I mean, clearly, like the visual of you, in someone's shared lab that they're loaning you a little bit of bench space is incredible. But how did you go about bringing in other collaborators to help you along the journey?
Yeah, so I knew that it was very unlikely that I was going to make progress on on my own as this like, third year medical student who's like, I just sat in on lab meetings to try to understand how people did science. And it was unlikely that I was going to make the progress on my own. So you're right. I focused actually most of my attention on creating an organization called the Castleman Disease Collaborative Network, with the goal of getting all the physicians, researchers, and patients together, connected. And though Castleman's is rare and not that many people have heard about it, there are actually about 5,000 patients diagnosed each year in the US. Okay. So it's actually about as common as ALS, certainly a lot less awareness for it. And as a result, that meant that there were a lot of patients all over the place. There actually are a lot of doctors that have treated patients. There just was no community for them to coalesce around. Mm-hmm. And so we created that community, and we actually took this really kind of radical approach to research, where rather than just raising money and then kind of hoping the right researcher applies for the right project at the right time, we actually went to that community. It took about 1.5 years to do this, but we went to physicians, researchers, and patients, connected them online, began to have them communicate online, and then started crowdsourcing among that community what research should be done, what research questions would be meaningful for patients. And once we figured out what we needed to do, then we went out and we recruited the best people in the world to do it. So it's a big flip to go from, hey, who wants my money? And then you wait for the researchers to apply to you to say, what should we do? And then go find them. And that's been just revolutionary for us.
Yeah. Have you extended any of the, kind of things that you learned for yourself in terms of, high frequency testing or anything like that? Have you now tried to expand that circle? Are more people trying to do what you did?
Yeah, so we've tried in a couple ways. And because you can imagine, here I am, it's been 6.5 years, over 6.5 years since my last relapse because of this system, right? That because of the community of physicians, researchers, and patients, because of the repurposing approach, and I'm alive today because of it. And so you can imagine that someone like me who just, I wake up every morning, I'm just like, I can't believe it. People ask me if what happened in the past felt like a nightmare and this is reality. And it's actually the opposite. What happened in the past feels like reality and this feels like a dream. And so for someone like myself, you can imagine that all I can think about is, how can this be shared with other people, right? Like I feel like I owe the universe to be able to share, what's enabled me to be here today. And so the couple ways that we've done that, the first is by partnering with large advocacy umbrella organizations. So the Chan Zuckerberg Initiative and Global Genes, which is a great advocacy organization in the rare disease space, to really try to map out the entire approach we've taken to accelerating research for Castleman. So we call it the collaborative network approach, mapping that out, and then partnering with Chan Zuckerberg Initiative to create tools that could actually be shared and scaled with other rare diseases. We did a lot of bootstrapping to create our own online tools to just basically make it work for free. But we've worked now with CZI to create tools that could actually be scaled beyond Castleman's. In addition to that, the other way that I'm kind of trying to evangelize for this is through my book. It's through Chasing My Cure, which came out about 1 year ago. And it's hopefully sharing the power of collaboration, but also sharing the importance of looking at old drugs in new ways. There are over 2,000 drugs that are already FDA approved for something. And many of those have not been systematically and fully evaluated for all the potential indications that they could work in.
Yeah, it's interesting. There has been this explosion of companies using artificial intelligence to try and glean more from published data. But in general, you realize that the drugs that have been developed over decades all had a purpose of hitting a particular target and doing a specific biological thing. And the more that we learn about biology of various diseases, the more likelihood there are that, certain switches in certain ways are actually pretty consistent across medicine and across different diseases. So there's certainly, it's another kind of niche of opportunity within what the biopharmaceutical industry can do and do well. And a lot of times it can be unlocked with biology by understanding exactly what the drugs do, or, conversely, understanding the actual disease mechanisms that we didn't really understand before. And you have a really good perspective on this because you were both a patient and you were actually, a doctor. You basically had both sides of this perspective while people were attempting to diagnose you and really getting nowhere. Right. So do you feel that we're fundamentally getting a little bit better in the rare disease space of figuring out what kind of things that people have? Or or is it so biologically cloudy that, we're doing the best we can with tools that aren't all that great?
So I think that it's more the former. I think that, as you know, there's just been an explosion of technologies and techniques to understand biology in ways that we never could before. We do a lot of single cell RNA sequencing in our lab, and just the concept that we can look at, single cell resolution to what is that cell doing? How is it behaving? What cytokines is that cell producing? Is really just kind of mind blowing, frankly. And so I think the tools that are available today enable better biological understanding than ever before. And then the question becomes, well, what do you do when you understand the biology a little bit better? It's one thing to say, okay, this is the gene that caused the disease, or this is the protein that's causing, your cytokine storm. But then what do you do with it? And I think that the what do you do with it is a little bit harder. Well, it's not a little bit harder, it's a lot harder. And so, it's how do you develop a drug that's going to actually reverse the thing that you just figured out?
Right.
And or identify an existing drug to reverse the thing that you just figured out as the problem. And unfortunately, when you think about the existing drugs, so drug repurposing approach, there are actually very few incentives that exist to actually make this happen at any sort of a scale, at least for drugs that are generic. For drugs that are still on patent, repurposing is certainly, it's more financially viable because there's more of an incentive to actually do the work to figure out what other indications could be used for. But once something becomes generic, it is so much more difficult to harness the resources to figure out what are the other diseases that could work in, because there isn't the financial incentive that exists. So that's a challenge. And then, as we know, in the rare disease space, where cellular and gene therapies are really so exciting, and we're all so hopeful will deliver kind of on the promise that we know that they can. Even cellular and gene therapies, it's going to take time for all of these, there's 7,000 rare diseases, and we see how hard it is to get every one of these gene therapies approved. And oftentimes, and almost always, they're approved for one particular variant of one particular rare disease. And so I think that we need to think about scale. And so we're understanding biology better than ever, but now we've got to figure out, how do we translate that biology into therapeutics?
Right. So in your case, the assays and things that you would run on yourself, you would figure it out that it was affecting the mTOR pathway, if I remember correctly, right? Yeah. So can you take us through a little bit of, A, how did you get there? And B, like, you get there and you have the pathway. So, then what did you, how do you get from, I figured out the pathway, can I find a drug? And once I think I might have found a drug, how do you actually get it? How did that transition work to, I think I found something, to how do I give this to myself?
Sure. So I'll step back to before I found it, because the second part, which you're asking about, I think was maybe almost a little bit easier than the first part, which was identifying the therapeutic target. And and so for me, I had my 5th deadly flare in December of 2013, and at that time I just finished medical school, and I had enrolled at Wharton for business school, because I learned while I was chasing my cure that some of the biggest hurdles for making progress were actually issues of organization, strategy. These are things that you guys know so well. It's not a science problem always. In fact, many times it's a human problem or it's an organizational problem. And so I enrolled at Wharton, and I was kind of almost a semester in when I had this 5th deadly relapse. And at that stage, I'd just gotten engaged to my girlfriend, Caitlin, now fiance. And all I wanted to do was to make it to May 24th, 2014. That was all I could think about. Can I find a drug that can help me to make it to May 24th, 2014? If I made it to May 25th, sounds great. But I just needed to make it to May 24th. And
I take it that was your wedding date?
That's my wedding date, exactly. Yeah. Yes, I should have explained. You're like, what is it about May 24th? Yes, that was.
We got it.
That was the date that we had set, and so I spent a month in the hospital, and I got really, really sick. I nearly died for the 5th time, and needed a combination of 7 different chemotherapies again to save my life, and just kind of miraculously made it thanks to chemo. But at this stage, I knew I needed to find something, and I knew I was running out of time if I wanted to make it to our wedding day. And so once I got out of the hospital, I just was 100% all in on trying to find something. And so the first set of data that I started looking at was actually just existing data in my medical records. So my doctors have been running these cytokine panels where they measured the same 13 cytokines every couple of months. And so I went back to these cytokine panels, and what I found was that during my flare, so Castleman's is a cytokine storm disorder, very similar to what we see in COVID-19. In the sickest COVID patients, it's a cytokine storm that if you look in the blood of a really, really sick COVID patient at the cytokines that are elevated, it's almost indistinguishable from a multicenter Castleman disease patient's cytokine profile.
Okay.
So it's this incredible multi-organ, hyperinflammatory, you're on a ventilator, it's bad. Yeah. But so during my flares, all of my cytokines were through the roof. It was really, really bad. But what I found from these panels was that actually in the weeks and the months leading up to my relapse, there were 2 cytokines that began to rise before other symptoms arose. And one is a cytokine that is a marker of activated T cells called soluble IL-2 receptor. The other one is vascular endothelial growth factor, which many of us know because there are drugs that target VEGF, but VEGF is important for blood vessel development. And so this was kind of the first set of like, well, maybe maybe T cells and VEGF are involved in some way. And so fortunately, I also, during my relapse, I had been storing serum and peripheral blood cells on myself during that relapse and leading up to it. And so I had the serum and I had the cells, and I ran proteomics on the serum, and I confirmed that in fact VEGF and this marker of T cell activation were 2 of the highest. When we looked at, rather than just looking at 13 cytokines, we looked across like thousands of analytes, they were way up at the top, which was helpful. And then I looked at my cells, and I found that actually my T cells were highly activated, like out of control activated.
Yep.
So now I kind of confirmed these 2 things, went back to the proteomics data and ran what's called pathway analyses. So you're just asking the data, are there any sort of signaling pathways that maybe you could back into based on the proteins in the blood? Are there things that could kind of piece this massive picture together? And 3 different tools predicted the mTOR pathway. They predicted a lot of other stuff too. And that's what's important is you get a lot of false positives, like malaria came up as part of the proteomics analysis. I don't think I ever had malaria, but like, that's what happens when you do these analyses. You get a bunch of things. And then you have to say, well,
you did grow up in the South.
That's right. Not that far south, but yes, my parents are from Trinidad in the Caribbean. So maybe somehow there was transmission. But so you get all these results, and then you don't know what to do with them. But the helpful thing was that the mTOR pathway was the only result that came up across the 3 different pathway tools. It made us feel like there was something there. And of course, it could have still been random chance, right? But that made me feel like maybe we were onto something. And then I remembered back to med school that the mTOR pathway is critical for T cell activation and for VEGF expression. So it fit with kind of what we had been seeing previously. And then the best part there's 2 great things about mTOR. One great thing about mTOR is that there's a really, really simple test you can do. Even I could do it. A really simple test to be able to determine whether mTOR is active in tissue. And so I did that on my lymph node tissue, and I did it on normal tissue, and it was just basically black and white. It was like, I'll never forget. I didn't even have to look under the microscope because it was so dark that I could see it with just the naked eye.
You could see the stain with your eye. Yeah.
Oh my gosh. So, and then I looked under the microscope and I was even more like, oh my gosh. So mTOR clearly through the roof. So that was great. It's easy to do this assay. But then the next best thing about mTOR is that there are a number of drugs that are already FDA approved that inhibit mTOR. In fact, one of them, sirolimus, has been around for over 30 years, has a really well-established safety profile. It's used for kidney transplantation, so people take it every day for decades. So you know that it can be used long-term. It's not a drug that's usually, a 5-day course. This is a drug that can be used for years and years. And so now I get to kind of your question, which is, okay, mTOR, now what? And sorry for that long answer.
It's helpful.
I found myself smiling when you started asking me the question, just because it brings back such good memories that I had to go there. But so once we now had this target, I shared the data with 2 of my doctors, and I said, we're out of options. mTOR seems to be up. What do we think about using an mTOR inhibitor? And they both agreed that in the absence of other options, like it's worth a try. And so fortunately, sirolimus is generic, so it's a fairly cheap drug. It's It's more expensive than others, but certainly cheaper than most of the chemotherapy and most of the monoclonal antibodies I was receiving up until that point. And so my insurance company didn't even ask a question. They just, they approved
Yeah.
it. So the drug had never been used in my disease before in the world. But I guess because the insurance company, didn't see alternatives, they approved it. And I started taking it within a couple of weeks of that experiment. And as I shared earlier, it's been over 6.5 years that I've been in remission.
Well, and that's also a good example of I feel like they're fairly rare, but the insurance company recognizing the value of a potential therapeutic over the cost of another, near-death hospital stay.
Yes, exactly. And I think you're right. I think that it probably was, and they're very good at weighing, the cost and benefit. And I think that was exactly right. My hospitalizations are close to $1 million when you're in critical condition and you're, spending weeks on life support. They don't want to spend that. And so even if there's no data to support this mTOR inhibitor, spending the X $1,000s a year is going to be potentially major cost savings over hospitalization.
Right.
Mm-hmm. So we have a question from the audience that I think fits in now, which is, what are some of the bottlenecks on availability of past drug data and either drug sample access or access to compounds? Are these some of the most critical things to fix to make doing this for other ailments easier? Said a different way, what are some of the barriers to doing more of this type of analyses on patients and making this type of research more effective?
Yeah, I think that the first thing that was mentioned is spot on. So as you know, there's a lot of data that never gets published. There's a lot of data on drug might have worked in this condition, but the safety profile wasn't good enough. So we just, moved on, or vice versa, it didn't work and the safety profile was really bad. There's a lot of combinations and a lot of data that just never get published. And so what that means is that when people like me are looking at sirolimus or any drug for a condition, we have a blind spot, right? Like someone in the world somewhere that data exists, but it's a blind spot for me or any other scientist. And so we have to try to regenerate that data, or maybe even worse, we don't regenerate the data and we make a mistake that could have been avoided if we had the data. So access to existing data is a huge, huge bottleneck. I think the other really, really big impediment is really the fact that there's so little financial incentive for this sort of work. It's actually not that hard to get a compound library, to get basically, a small amount of each of these FDA approved drugs and to do high throughput drug screening. But high throughput drug screening, well, first off, it has a really, really good track record at finding a first disease to go after, like a first indication. Basically, you figure out a drug target, you do high throughput screens, you figure out this compound works really well on this target. So that works well for that. But historically, high throughput drug screening has not been effective really at all in saying, what else could it be used for? It's actually, unfortunately, there aren't many successes. There's a lot of uses, and I'm a big proponent of it because anytime it works, or if it does work, that's amazing, and we should keep shooting on goal. But unfortunately, it has not been nearly as effective as I think all of us really hoped it would be. So it's not so much a problem of not being able to get the compound. I think it's more that if you find out that a compound is exciting, and there's actually a lot of published papers where it's like, this pathway could be helpful in this disease, and it looks promising. And then, so I'm a basic scientist now. I run a research lab where I do that kind of work. And then many of my colleagues just kind of, drop the mic and move on to the next thing. And literally it just falls through the cracks. It's one of the, millions or, whatever number of papers get published. And so and it's because there's no financial incentive. Now, if it was a new molecule that had this new activity, I can promise you that basic scientist is going to keep pursuing it. The university is going to pursue it. The companies are going to pursue it. But because it's just an interesting finding, it gets published, and then I don't think it gets acted on a lot of the times.
Yeah. There's a lot of institutional knowledge that never really, gets put out to the public. There are teams of people across biopharma that probably think, oh, we were going to try this drug for this, and we thought about using it here, but nobody, science has never accepted the paper of stuff we thought about, but ultimately kind of didn't do, even though we were mildly sure that it might have worked. Right.
Well,
So they're go ahead. Yeah.
And I think COVID is a good example, right, of a situation where all of a sudden we're dusting off the old libraries, and we're looking, and we're like, what might treat, and does it, and can we test it, and can we do even small trials that are not placebo controlled? It is literally throwing everything at this new disease. And so it almost takes something like a global pandemic to reset and have us think, okay, if we're doing this for something that is obviously as broad and widespread as coronavirus, then what else have we missed, or what else can we repurpose and look at?
Yeah,
I totally agree.
so yep. And so your network now you've now garnered enough support to be doing 2 randomly controlled trials, right? You not only have one for sirolimus, but another for BCG, right? So your network for Castleman now has 2 trials underway, correct?
There's no BCG trial going on in Castleman's. No. But yes, we are doing a trial. Unless if you're, it would be amazing if you're like, letting me know about the study. So we're not doing it, but maybe there is another study going on that I'm just not aware of. And that would be amazing if you're, let me know about that. But so yes, we are doing a trial of sirolimus. The drug that I'm on, we're giving to other Castleman's patients through a clinical trial. It's only being given to patients who fail to respond. I mentioned that drug earlier. It's siltuximab. It's an IL-6 blocker that works in about 1/3 of patients. That went on to get approval for idiopathic multicentric Castleman disease. So to get into our trial, you have to fail the IL-6 blocker.
Okay.
Then you can get sirolimus. And
Okay.
I think that there's an interesting lesson here. And of course, New York Bio audience, you guys are going to know this as well as anyone, but the importance of thinking about study design when you think about your disease. And so let's think about COVID for a second, where over 90%, maybe over 95% of individuals who get infected, they're going to get better. They're going to survive. It's a small fraction that will die, and that's without therapies, right? And so if you do an open label trial, which means that you're just giving the drug to people, you're not giving placebo, and over 90% of people survive, you have no idea if it was because of the drug or if it was because they were just going to survive anyway. In Castleman disease, you can get away with open label studies because there are no reported cases of anyone ever having idiopathic multicentric Castleman disease that has spontaneously improved. No one. So you will progress until you die. And so it's like pancreatic cancer. You can do open label trials in pancreatic cancer because unfortunately the natural history is that people will get worse until they die. And so we can get away with open label studies because in patients like cases like Castleman's and pancreatic cancer, it's actually really hard to convince patients to join a trial where there's a placebo arm when there's such a bad outcome. And I understand that. I mean, as a patient, I can appreciate that. And so we can get away with open label studies, and there actually have been randomized controlled trials like the one I mentioned, where 0% of the people who got placebo improved. And so once you do one of those RCTs and 0% improve, you don't need to keep doing those placebo controlled trials.
Right.
But COVID is
it's almost
opposite.
Unethical to do so.
Exactly. And so COVID is the opposite. Not everyone, almost everyone gets better on placebo, right? And so the key, you're trying to distinguish that small percentage that wouldn't have gotten better. And as a result, you just have to do randomized controlled trials if you want to get any sense for efficacy.
Yeah.
Yep. So we had another question that I don't want to derail us from COVID because I want to spend some more time here, but we had a question about the ethics around basically treating yourself as around testing on yourself, right? And it's obviously you're in overtime, so it's obviously a challenging situation. But can you talk a little bit about just the scenarios around research on yourself or on individual patients, and either how you navigated those things or how we can do a better job of navigating those challenges?
Yeah, it's really complicated because, as you said, it wasn't just testing on myself; it was also doing experiments on my samples. And so, So in order to do that, I had to have an IRB approved protocol, and I was listed as an investigator on that protocol. And of course, I also disclosed to the IRB that I would be one of the patients on the protocol. And so I think that in science and in medicine, it's really all just about disclosure. I mean, I think that obviously sharing with them, they had questions, but we came up with a plan, a risk management plan to make sure that as much as we could, it was de-identified as much as you possibly can. There's obviously challenges in doing that. But really the main thing that we did that was important for this, and I think important just for the scientific integrity of the work, and that's that it wasn't me working on my own. So my book's titled Chasing My Cure, but I think could not have been more of an incorrect title. It should have been Chasing Our Cure, because there actually was a whole team of people. And so, yes, I performed some of the critical experiments towards the end, but I always had other people help to analyze the data, because you can imagine that if you have a deadly disease and their clock is ticking, you might see things in the data that make you pick up a pattern that doesn't exist just because you're just so hopeful. Yeah, I didn't want that. I only wanted to find something real. I didn't want to find something not real, right? You want to find real results, but you could imagine someone like me could be biased to find not real results, right? And so as a result, I just shared the data and always relied on other people to interpret it. So that was one way that I kind of risk mitigated, was just to make sure I wasn't the only one looking at the data. And then as far as the kind of trying this drug on myself, I think that, the immune system is very complicated. So there are examples where you turn one thing off and that turns something else on, and you're in a worse situation than before. So just because I had this one stain that made my day when it was so dark, didn't necessarily mean that blocking that thing that was staining so positively was actually going to help me. I mean, it certainly could have hurt me. So I think the only way that scenario happens where I test that drug is a scenario where I've nearly died 5 times. So it's not like, oh, this is a bad disease, like maybe it'll be bad for me. It's like it already happened 5 times where there were no more options. So it wasn't like, okay, there's another drug we can try out that someone else had tried. And then thirdly, where there's like this future in front of me that I so desperately wanted that I kind of was willing to do whatever I needed to do to get to that future, which was May 24th, 2014. And so I think that it's this scenario that all 3 things lined up. And then I remember talking to my family about it. I was talking to Caitlin, my fiance, and to my family. And I remember them just being like wildly confident in the decision. And they're like, it's good. Yeah, absolutely. We trust you. And I was like, I wish I had this confidence. I wish I was like even a quarter as certain as you are that I'm onto something. I think I'm onto something, but they're like, oh yeah, we trust you. You're on it. And it's like, oh my gosh. So they trusted me. I didn't necessarily trust myself as much as they trusted me, but yeah, then it was just kind of taking a leap of faith.
Well, don't you think, part of that too is that you didn't want to disappoint them in your excitement, because they're part of your journey too?
Yeah, absolutely. I think that there's definitely an element of that. And, it wasn't even just me chasing my cure for me. It was, in many ways chasing my cure for them. Caitlin and I, wanting to be able to be married and have a family maybe one day, and spend more time with my family. So yeah, it was a team effort.
We actually had a question, and this is about, it's somewhat legal and political and talking about drug access, right? We had a question that says, should we change the law, and this would be federal law, to make it easier and better to patent old drugs for new disease? And I think that goes into the incentivizing R&D work on things that already exist.
So frankly, I think we have to do something. I think that the system just doesn't work the way it currently is, to be able to take old drugs and use them in new ways. Sirolimus is moving forward because of this, almost randomly work that randomly happened by me in my lab. And now I was able to garner the resources to do a trial, but that wasn't easy. And so this isn't like a very, easy to wash and repeat process. We have to do something. And I thought about that approach. I think that generic manufacturers would be, it would be like destroying business to be able to say like, okay, I know that it was generic and I know you were making it, but you have to stop now so that, a company can start making it, or a branded company would be able to start making it. But I think it's something that should be considered. I think that, that's a kind of an extreme approach. There are other approaches that maybe aren't as extreme, where you try to extend patent life for drugs that are still on patent. Of course, that's not going to address the 90% of drugs that are already generic. So it's kind of a small piece of the puzzle, but I do think we need to think through solutions. And I hope folks listening to this will be a part of thinking through new solutions.
Maybe something along the lines of what we did with the Orphan Drug Act, right? Where you're allowing access and availability to companies to develop commercial drugs for very small populations. And maybe that's a segue to that.
Yep. We also just had a question from someone about how to basically connect with the research network about a new therapeutic that people are developing that might have activity, which we can connect with you afterwards with more specifics. So I think part of this is realizing that, you know, there's a level of connectivity where the people who are working on certain things sometimes don't always know all of the places that they could try it. And it's one of the nice things where most of the people in this industry are doing what they're doing to help patients.
Mm-hmm.
And I think if people see new outlets for the research that they're working on that will benefit patients, they want to get that drug to those patients, right? They absolutely want to do that, whether it's within business scope or business plan or not. They want to see their therapies get to patients that they can help.
Yeah, so please do connect me. Yeah, I'd love that.
I will. I've asked him to send some additional info our way so we can do that. I didn't know if we could do it immediately live over a webinar, but we'll get there. So
as you learned, we're not that technological savvy.
You guys are great.
For those of you in the audience, we were not as wonderful technically this morning as we could have. So we managed to get things together before we went on, but just thanks to our crack production team for getting us on the air on time. I did want to get to a little bit to thinking about how you basically took some of the lessons learned from the Castleman's Disease Network and started to apply them to COVID-19, right? Because you obviously, did an amazing job of being able to connect people doing various types of research, connect people who were able to put together these clinical trials. So what type of things, how did you basically come up with the idea for, I believe it's called Corona, right? And and how did you get it to go live so quickly and so efficiently?
You may remember, or I'm sure you remember, Friday, March 13th, right before kind of the U.S. shutdown. That was the kind of last day before there was major nationwide shutdown, and I was driving down with my wife and daughter. So my wife and I had a daughter 2 years ago, Amelia, and the three of us were driving down to North Carolina to see my family because we knew shutdown was about to happen. And actually my brother-in-law is quite ill. So we wanted to see him and the family. And it was late that night, and I was listening to the radio, and I'm hearing about cytokine storms and how interleukin-6 may be important in COVID, and also hearing about drugs being used off label, being repurposed. And I remember thinking to myself, and I'm like, man, I really hope that some researchers out there really pay attention to the cytokine storm research we've been doing. So I run this center I mentioned, the Center for Cytokine Storm Treatment and Laboratory at Penn. And I said, I hope they follow our cytokine storm research. I hope they really dig through it. We've been doing this for the last 8 years. Hopefully they dig through the cytokine storm research. And then I hope that they also, kind of follow our drug repurposing approach. We've really had this really clear framework and stepwise approach to drug repurposing. I hope they follow that. And man, I really wish that, that I or my lab or center could help, but, we don't do COVID, right? We've never studied COVID-19 before.
Right.
We study cytokine storms. And then like 30 seconds later, I was like, wait a minute. Like, of course we don't study COVID. COVID didn't exist 4 months ago.
It didn't exist.
But we study the closest thing to it, and we study cytokine storms, and maybe we should just do it. And you might remember right around that time there was this like Big Bird meme, and it was Big Bird on a horse, and it says "We ride at dawn." And I don't know if you saw that, but anyway, I sent that to my team, and I sent it to the team, and I was like, we're taking on COVID. We ride at dawn, and of course, Big Bird has extra special meaning because I have a 2-year-old. So there's extra, special connection to Big Bird. But yeah, I mean, we decided, we ride at dawn, and we're gonna go all in on COVID. And so I redirected half of my lab to studying COVID, to reanalyzing data on COVID samples, and also searching for existing drugs that could be repurposed. So that's one part of it, but really the massive effort has been recruiting medical students and other volunteers from around the country to actually go through the entire published literature on COVID, which is about 2,000 new papers a week. But it's
Yeah.
the entire published literature and pulling out every time a drug is given to a human in real life. So every time a drug is reported to be given to a human, pulling data out on that drug and providing a central repository. It's the only repository that I'm aware of all drugs that have been given to COVID-19 patients worldwide. We found over 250 different drugs have been given to COVID-19 patients. And, you know, we hear about the same 3 drugs every day in the news, right? But actually, there have been over 250 different drugs that have been used. And now there's a long tail. The top 20 drugs have been used a lot, and the remaining 230 plus drugs have been used in very small numbers. But the point of this effort is really a couplefold. One is to say, if we're going to try to repurpose drugs for COVID, we need to keep track of our denominator. We need to know all of the drugs that have been tried, because maybe the drug that's been used 10 times needs to be used 10,000 times. Maybe it actually, that's the drug. And so it's tracking all of the drugs so that we don't lose anything, but it's also, frankly, looking at the drugs at the top of the list that are used frequently and really diving into the data. Earlier, you heard me get on a soapbox about why it's so important to do randomized controlled trials for COVID, because the natural history is that most people improve. And so Corona is this effort to centralize the data all in one place. And we've made it freely and publicly available. It's the website is cdcn.org/corona, and anyone can access the data, and it's a really easy to use format. And so if you work for a biopharmaceutical company, check out the website, see if one of your compounds is already being used. You may not be aware that it's being used for COVID. You can immediately get data on the use of that drug.
That's fantastic. I can't get the Big Bird image out of my head, but
I'll
for
share the
everybody
meme with you
listening
afterwards. But yeah, we write it.
Oh
down.
no, I'll find it, I'm sure. So for everyone afterwards, this will I'm gonna share the link, and I'll find the meme too. I'll share the link and the meme via both Twitter and LinkedIn. So David, I'll tag you and look for retweets on that one. You aren't gonna get away from this one. You know, we had another question here is saying, is your work being applied to basically post-COVID inflammatory responses where major organs are being attacked? So are you actually seeing some of the fruits of what you've done be applied in the COVID space?
Yeah, so very much so. There are a number of drugs that were developed for Castleman disease or that are part of our pipeline for studying Castleman's that are very much at the forefront right now. There was a pretty big negative, and that's a drug called tocilizumab that was actually developed for Castleman disease back in the 90s. It was developed and approved for Castleman's in Japan by a friend of mine, actually, Kazu Yoshizaki. Kazu developed it, and I guess it's kind of related to my story. He actually tested it on himself to prove that it was safe before he gave it to other patients in a phase 1 trial. And when I asked Kazu, I said, Kazu, I heard you gave tocilizumab to yourself to prove that it was safe. He said, no, I didn't give it to myself. The nurse, she gave it to me. so Kazu discovered that interleukin-6 was a target in Castleman's. Okay. He developed tocilizumab. He tested it on himself, proved that it was safe. Then it went on to phase 1 trials, got approved for Castleman's in Japan. You guys recognize the name tocilizumab because it's used across the United States, particularly in cell therapy and also in gene therapy when people have cytokine storms because of the cellular and gene therapy. And so tocilizumab, Castleman's drug being repurposed here in the US. It's approved for rheumatoid arthritis here in the US. But again, the history is it was a Japanese drug for Castleman's. That actually was a drug that we were really optimistic about based on the overlap between Castleman's and COVID. And then the RCT read out a few weeks ago that it was not effective compared to placebo. This is a great example where there were so many open label studies where I was talking to doctors in China and in Italy, across the United States, that were swearing by tocilizumab. This drug saved my patient's life. They just, walked out of the ICU. And then you do an RCT and you find out that people who got placebo did just as well. And so I guess I just shared a notable failure. But I think that it's important for us to pay attention to what works and what doesn't work. Let's not keep failing. Let's make sure that we're learning from our failures. And we have a paper under review right now where there's a few more drug targets, all thinking about the immune system and the host response. When you think about fighting a virus, you can either target the virus directly, or the two approaches we like to think about within the host immune response is you can either immunostimulate, so boost up too weak of a response, or you can immunosuppress, turn down too strong of a response. And what it looks like is that we really need to be hitting all three of those things, but we only stimulate if it's early, and we suppress if it's late in the course. And so Dex is a great example where dexamethasone looks like it actually harms you if you take it too early in your course, whereas it's wildly effective if you take it late in your course. And so, and I suspect that drugs like interferon beta, which are coming down the pipeline, which seem to be effective early, I suspect they actually would be harmful if you give them too late. And so it's really about trying to find, again, this is an analogy that is very much relevant because I have a 2-year-old, but it's kind of like a Goldilocks phenomenon where it can't be too hot of an immune response, can't be too strong, but it really can't be too weak of an immune response.
Yeah. So Ed Yong recently wrote a great article about why immunology is just so incredibly complicated, right? You know, it's I think we're learning more and more about it with the advent of immunotherapies. And, you know, it's interesting with COVID and with your story, anybody who's paid attention to cellular therapies, T-cell therapies, rare gene therapies is reasonably familiar with cytokine storms as a side effect, right? So it seems like this is something that, you know, given how many new therapies are being developed in those classes, I think this is an area of study and an area of safety that we're going to, you know, fortunately or unfortunately need to be much more familiar with in the coming decades. It's going to, I think, unfortunately be more commonplace, but hopefully the things that we're learning about it now adds to a much more applicable level of either diagnosis before they happen or safety after they do.
Yeah, I couldn't agree with you more. I think that it's really about something we were talking about early on in the conversation about how we can't think about this just kind of one disease at a time. We have to learn principles about cytokine storms, immune hyperactivation from X condition, and then we need to think about how can that apply to Y condition. And we need to you know, I think that we as scientists and folks in biopharmaceutical industry, we're very good at like kind of running down our lane full speed. But I think that from a scientific perspective, COVID's a great example where, like, myself included, but everyone has kind of run into the COVID lane. And we've proven that we can, right? We've proven that actually, and of course, there have been some notable issues in science since COVID emerged, but we've proven that we can apply what we know from our disease to another condition. And so now it's to Jennifer's point earlier that like we need to do that. We need to start sharing across diseases, and maybe we can create some silver linings from COVID-19. Maybe, I mean, COVID has been uniformly in every way unequivocally bad, right? It's been bad. But if something positive can come from it, maybe people collaborate more, maybe they share data more frequently, maybe drug repurposing can be used more frequently. I mean, I think that would be amazing.
David, we have time for one, unfortunately, only one last question. I think we could definitely go.
We have a perfect one. We have a perfect one.
Yes.
We do. So
yes. And so we've talked a lot about your focus, and you're writing at Dawn, and in your book, you talk about your singular focus, right, on things. But one part of the journey is, it's a question as a family advocate, the journey can be overwhelming and emotional. We hear these stories and they're amazing, but they seem so out of reach to us. What is your advice in making the story heard from the family advocates in the journey?
Yeah, it's such a great question. So I have to say, for me, it felt so out of reach for me. I mean, I nearly died 4 times before I started doing anything proactive. For 4 times, I was like, what am I actually going to do? Someone else has got it. Like, I can't really make a difference. I was very reluctant to dive into it because it did feel so overwhelming. And I was a medical student, so I can appreciate that, you know, not being a medical student, that would be really, really complicated. But I think what I've learned is that my story is a reflection of what happens when physicians, researchers, patients, and advocates work together. You don't need to be all 4 of those people. You can just be one of them. But if you can get the other 3 people together, then you can make incredible progress. You just won't be able to make it on your own. I mean, I said earlier, my book's got the wrong title. It should not be Chasing My Cure. It's Chasing Our Cures, because it couldn't have worked if it was just me. So for that person that's asking that question, it's not that, you know, can you do it on your own? Because none of us can do it on our own. It's, can you find those 4 people? Can you be one of them? And can you find the other 3 to really, really change things for your disease?
We are out of time. Thank you so much for joining us. This was fantastic. I know our audience learned a lot, and hopefully now they have a lot of other things to think about as they go about their work on R&D in other areas. Thank you so much. And then again, thanks to our friends at Goodwin for sponsoring our breakfast series.
Thanks so much, Jennifer.
Thank you so much,
Karen.
everybody. David, thank you. Have a great day.
Thanks so much. Bye,
Bye.
guys.
Thank you.
Thank you for tuning in to NewYorkBIO's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on NewYorkBIO, please visit us at www.newyorkbio.org.